Hi, my name is Scott Berry. I'm a biostatistician at Berry Consultants, and I'm going to walk through examples of utility functions, modeling efficacy and safety, efficacy and tolerability using utility functions. I'll show you several examples of these and good practices. So the first example I'm going to talk about is a trial in stroke. The endpoint at 90 days in this example is the modified Rankin score. You can see on the table on the right the 7 levels of the modified Rankin score. It ranges from level 0, which is no symptoms, perfect status, neurological status, down to level 6, which is dead. And you can see that the levels vary 1, 2, 3, 4, 5, and 6. An interesting thing about this is the number does not classify the relative difference in the states. It's just a classifier. You could label these A through G as the label for the level. The question comes up about how do we analyze this? What is the statistical analysis of this particular endpoint? And there are a number of different ways to analyze this. We had a particular trial where this was the endpoint, and the comment was made that let's analyze it where we lump in 0, 1, and 2 as one particular dichotomous category, and then 3, 4, 5, 6 as a separate. And so we dichotomize it. The graph on the right shows you the relative utility of those 7 states. It's equal to be 0, 1, and 2, and equal to be 3, 4, 5, and 6. And the comment was made is let's analyze it mathematically this way. We separate them mathematically, and then when the trial's over, we'll think about clinical importance in the trial. I didn't like this. I felt like this was bad science. It separates the statistical analysis from clinical benefit. So I feel like this is a statistical mathematical test that doesn't tie in well to the design, to the clinical question. As clinical trials are becoming more innovative, we can't do this that we wait till the end of the trial to address the important issues. Adaptive trials, response adaptive randomization, enrichment, I'll show you some examples of these. You need to be able to classify that upfront and prospectively so that the design does smart things during the course of it. So we need to address these questions up front. Utility functions are very nice ways to do that. So here is an example trial. It's called the DAWN trial. It was a trial in a clot clearing device for post-stroke. Now, this is approved by the FDA within 8 hours, but this was a trial looking at subjects beyond that period. It actually enrolled between 6 and 24 hours, and clinically it looked for a mismatch. The primary endpoint in the trial is 90-day modified Rankin score. We propose the primary analysis to be a utility weighting of those 7 outcomes, in part because of the complexity of the design itself. The design had enrichment steps to it where we might stop enrolling certain types of strokes if the device was not beneficial for that. This was an adaptive enrichment step. There was also early stopping for futility and success in the trial. These are prospectively defined rules that we need to have set up. So we can't do this aspect of, oh, let's do a mathematical test and think hard about it afterwards. We need to incorporate that prospectively up front. So in this trial, we created a utility function. So what is a utility function? A utility function is a mapping from a set of parameters. So in this example, it could be the probability of these 7 outcomes, maps it to the real line. What is nice about this is it maps what could be multiple endpoints into a single measure. This utility measure on the real line doesn't have units. It's not in terms of dollars or anything else. It's a relative value of doses or arms that we can directly compare on that measure of utility. So let's look at that example of the DAWN trial. On the graph on your right side over here, you'll see the dichotomous utility is the red, where we value 0, 1, and 2 the same. In this one, the blue dots represent the utility score that was used of each particular outcome. The utility weighted MRS, and you can read about it in the citation here. You can see the value of these within the 0, 1, and 2 have monotonically decreasing states, but a 2 has different utility than a 1. It's a worse clinical state, and 1 is worse than 0, and the height of these is relatively important. You can see the drops from 0, 1, and 2 are relatively smaller than the drop from 3 to 4 or 4 to 5, which clinically is a more important drop. This is the utility that was used in the Dawn trial for analyses. The Dawn trial ended, it stopped for success early in the trial. It never enriched, so it kept enrolling the large population. You can see here both this figure and table from the publication of the paper shows you the data. It shows the relative number of 0s through 6s on the active device arm and the control arm. And then the bottom table shows the primary analysis of the utility weighted MRS. The posterior probability of superiority of the active arm, which was greater than 0.99, and the relative difference on the utility scale. So you can see what a primary analysis of a utility looked like. It was really important in this trial to incorporate the relative value of these 6 states so that we incorporated the efficacy as well as safety. The potential that we could create more negative outcomes is directly captured by the endpoint in the trial. Now, utility functions, that's an example where we have these 7 levels of a single modified Rankin score. Other common uses of utility functions are to incorporate efficacy and safety or efficacy and tolerability directly in a single measure. And again, as we do innovative designs and we're doing adaptive designs, we can incorporate all of these into a single endpoint so the adaptive design does smart adaptations in the course of the trial. We're used to doing things like an ED90, which is in a way a poor man's utility function to say, oh, let's get 90% of the efficacy and not push it too high and target that because we don't know about tolerability and safety. This is a way of directly capturing safety and tolerability in your endpoint of interest, the utility. Let's look at an example of this. So this is an example efficacy curve over a range of doses. You can see these 10 doses across there, and the black line on this curve represents the probability of a positive efficacy outcome. Let's use an example of a treatment for insomnia. That this represents the proportion of patients that have a good night's sleep after taking the drug. That's efficacy of the drug. You can see the yellow line here represents the ED90, the effective dose 90. Whether that's the right dose depends entirely on the other endpoints, the tolerability. So let's look at potential tolerability curves. So I've added here 3 different colored curves that represent the probability of a tolerability issue that arises. In this example, it's easy to think about that you're overly fatigued the next morning after taking that particular treatment. There's a blue curve here and a green curve here and a red curve here representing the 3 different levels that could be for the different doses. You would love it to be the green curve because that has a less chance of tolerability. In this example, in the green curve, you would push the dose higher because you can get more efficacy with less relative tolerability, where the light blue curve, you would want to push the dose down farther. You still get efficacy, but you get less tolerability. The ED90 is the same dose regardless. So we could attach utility functions to the relative weight of efficacy and tolerability in this example. Let's look at several examples of this. We'll use a simple utility function, and as you can see here, the utility function is the differential between the placebo and each dose on efficacy, subtracting off the added probability of a tolerability event. So we're really looking at the difference between the black curve and each of the tolerability curves with an equal weighting of efficacy and tolerability. You could imagine a coefficient of tolerability making them more important or less important, or more complicated functions. But this is a simple function really of looking at the space between the two curves is the relative value or utility of a dose. So with that utility function, the graph on the right side of the slide here shows the utility of each of the doses using the utility function we described in equal weighting of efficacy and tolerability. On the bottom of the graph, on the x-axis, you can see the maximum utility dose accounting for both of these endpoints. And in the case of the blue to the green, you can see the higher the dose, the more utility when tolerability is less. And these are all different than the ED90. So this is a really nice tool for phase 2 trials, for adaptive trials, to incorporate efficacy and safety or tolerability into a single endpoint. I'll describe an example of a phase 2 trial that ran using this technology. The drug is Trulicity. Here is a reference you can look at to look at the design of the paper. It was a phase 2/3 seamless trial that had 7 doses in phase 2 with response adaptive randomization. During the course of the trial, every 2 weeks, the randomization probabilities for the 7 doses were updated to favor doses that were performing better. The way we judged better was based on 4 different endpoints all combined together into a single utility function that measured the therapeutic benefit to a patient of each particular dose. An algorithm selected the doses to seamlessly move to phase 3. It also spanned other phase 3 trials. Those doses selected by that utility function are now the marketed doses of Trulicity. The example in that trial, the utility functions are shown on the right of the slide. You can see how we combine together the utility of an efficacy endpoint, change from baseline in HbA1c relative to an active comparator. We combine heart rate and blood pressure and weight loss all into a single utility. The way we did that in this trial was by multiplying each of the 4 components you see on the right of your slide into a single utility function. And you can see, for example, if heart rate or blood pressure had too high of a difference from placebo, the relative value of the dose would be near 0. And we would go back to doses that were smaller, that had high efficacy, but had a better profile for safety or for weight loss. This was used in an adaptive trial in that particular example, and you can see the power of this within an algorithm. So to sum up what I've talked about, utility functions can be incredibly powerful tools for capturing multiple endpoints into a single function. Very powerful as we're looking at innovative designs, adaptations, algorithm-based decisions in this. Now, of course, the difficulty in these trials is creation of the utility function, eliciting that utility function for your particular scenario. This can be challenging, and to represent whose utility, are we looking at patients? Are we looking at financial? Are we looking at relative value of doses? But these are really the right scientific questions we should be addressing to drive the design into the right space. Thank you.